Because adaptations are completely dependent on the environment, it can be difficult sometimes for scientists to assess whether traits really are adaptations or not, or what traits might be an adaptation in one environment and then shift and not be an adaptation when you move to a different environment. So there are two ways that this can be determined. You can do this experimentally by manipulating a system, putting organisms into a new environment, seeing if things change. That can be very tricky, and there can often be many other compounding factors that make that difficult to assess. Additionally, what you can do is you can take many, many observations in the natural world. So rather than doing a manipulative experiment, you're just doing an observational experiment or making many observations to test a hypothesis. And so you can do both. And again, both have advantages and disadvantages. So let's look at an example of one where they did a little bit of both and we'll use it as kind of a test case for how we determine whether or not something is an advantage. So, is it better to have many small offspring or a few large offspring? And this gets to a central idea, and we're going to talk about this idea through much of the rest of the semester, is the idea of a trade-off. Okay? A trade-off means that there's going to be an advantage in one area, but that means of necessity you're going to have to give up something or it will be a disadvantage in another area. Number of offspring is a really good example of a trade-off. Organisms have a certain amount of energy that they can devote to reproducing. So for females, that's how much energy you can put into your eggs if you're laying eggs. And there are basically two strategies at the extreme and then everything in between. Now, ecologically, and you might be familiar with this, the result of this trade-off, we'll write it up here on the slide for you, means that there are two extremes to this. And in ecology, this is called a K strategy, and it's usually lowercase k, so let's get it back there, and the R strategy. And so basically that's what this experiment is looking at. So let's clarify that. A K strategy is you take your resources and you have lots and lots and lots of little tiny babies. Now, usually what that means is each individual is going to have a lower chance of survival. However, since you have many, many babies, even if they have a lower chance of survival, the odds are a few of them are going to make it. An R strategy is you invest your resources more heavily in each individual, which of necessity, because of this trade-off, means you're going to have fewer babies. So each baby has a better chance of surviving overall, but you have fewer babies in the environment. And in different niches, different strategies are going to be successful. So humans obviously are an R strategy species. We put lots and lots of resources into very few offspring, and that has worked very, very well in our environment, in our niche that we're filling. Many organisms that have complex behaviors and have to train and teach offspring and kind of show them how to survive, they need to be R strategists because they just can't do that with lots and lots of babies. On the other hand, a sea turtle, for instance, is an example of a vertebrate with a K strategy. They have lots of little tiny babies, and that maximizes their survival in their environment. But the mothers, the fathers don't teach or train or help the babies other than the initial input that the mother gives to the eggs. Okay, so those are basically the two things that we're looking at. And in this example, scientists were looking at guppies. And like most species, there's variety and diversity. And we can see this to some extent even in humans. So some humans are genetically programmed to have multiple offspring, twins or even triplets. There's maybe a very extreme example of this. You can look it up. Look this up. What is the record for live births? A human female, what's the most number of children that a human female has had? The number might actually surprise you, so Google it. But anyway, there is some genetic diversity even in humans. But in other organisms, we see a lot more variety and diversity. And in these guppies, there's a range. Now, what they noticed is that if guppies were living in the very upper regions of a stream where there were very few active predators, that they had very large offspring and had few of them. And that was best in that environment. Guppies in lower environment where there were much bigger active predators, they had very small babies and had lots of them. And so the hypothesis was that that is a adaptation. There has been natural selection acting on the diversity that's already there. And so in this example, people actually did these experiments where they found streams that they could move guppies into that they weren't there before, and they took guppies that had adapted to a lower part of the stream and they moved them to a new environment where there was no longer predation. And then they looked to see what was going to happen, and vice versa. Okay, so we take guppies that are adapted to a high predation environment in the lower part of the stream. We move them to an upper part of the stream where there's now no pressure, and then we watch to see what happens. Now, the prediction is that if this is an adaptation to that environment, that over time we should see a shift, as long as there's still diversity left, we should see a shift towards fewer and fewer offspring. And sure enough, that's what happened. So if they took guppies that had been raised in this high predation and pressure where there's still lots of variety and diversity, and they moved them into a low predation environment, pretty soon they were having fewer and fewer offspring. And so that is a good experimental test that demonstrates that, yes, indeed, this shift to a case strategy is an adaptation that they have gotten over time to that environment. And if we move them to a new environment, it's no longer an adaptation, and we see a shift in the phenotype. So that's a good example where they tested a trait that they thought was an adaptation and gathered data that said, yes, sure enough, it is. Okay? And not only that, but it allowed us to estimate and measure how fast natural selection works. And the answer to this question is complicated. But the short answer is that natural selection can be very, very rapid. And in fact, let's add one more term, which we didn't explicitly define when we first introduced the theory of natural selection, but we're going to define it now. Okay? And this is the term artificial selection. Now, I think there's a lot of misconceptions about artificial selection. Artificial selection is simply natural selection, but let me put it in here, artificial selection. So it's really just natural selection, but humans are determining the selective environment. So humans decide who gets to reproduce and who doesn't. And artificial selection is just a very, very strong form of natural selection with humans doing the decision making. And so when the selective force is very, very strong and very constant over time, natural selection can be very fast. So in these guppies, it was actually just a few generations, and they chose guppies because they reproduce very, very rapidly. There are many generations each year. And they were able to see in just a handful of generations a rapid shift towards the new adaptive trait. Okay, so natural selection can be very, very fast. Artificial selection is an example where just over a few generations, humans have had major changes on organisms. A good example is, well, really you can choose any domesticated crop or domesticated animal, and you can see the strong force of natural selection that humans have imposed on it via their choices. Dogs are a really great example. So over just a few hundred generations, right, over the last few hundred years, we have developed many, many different varieties and breeds of dogs. And there was some going on even before that. So humans have been selecting for certain traits in dogs really for thousands of years. But it's really only been over the last couple hundred of years that it's been very strong and very focused to develop certain breeds. So for example, if you look at the way the ancestors of Great Danes looked 200 years ago, you're going to see very, very different organisms than we have today. And because of humans' desire and interest in dogs as pets, there's been an incredible range of dogs, everything from the little Chihuahua to the Great Dane in size. We can look at coat color, we can look at behaviors, hunting behaviors, guarding behaviors that have all been selected for in different breeds of dogs. So that's an example of very, very fast, and we would call it artificial selection, but really it's just natural selection with humans being the main force in the environment. Now, here's one other experiment we're going to be looking at where it's manipulated, where we have the ability to control the environment and experiment much more so than we did in the guppy, where basically the only manipulation was we move them to an environment and then carefully watch what happens. This is a very famous experiment. It's been going on, I believe it's Michigan State, for now about 30 years. And this experiment started with one single colony of bacteria. Okay, so they started with that, and then as bacteria do, you let them reproduce, divide, and then you can separate them and put them in different environments, or put 12 different replicates in the exact same environment and look to see what the outcome is. And so it's a very nice experiment, and bacteria are ideal for this. They're not very complicated, but they're ideal for this because they reproduce so quickly. Under ideal conditions, you can get a single generation of bacteria in about half an hour. And so you can go through thousands and thousands of generations in just a couple of years. Okay, and so this is a long-running experiment where they've actually done many, many different tests, many different varieties and diversities. In fact, let's look this up. It's kind of a fun thing to look up. I forget the official name of it, but we can find it here. Long-running bacteria experiment. Okay, so it's called the E. coli long-term evolution experiment. They didn't pick a very memorable acronym. Maybe that's why I couldn't remember it. And they've gone up now. This is actually a little bit dated. They're actually higher than this, but they had 60,000 generations of bacteria. So they can look at what's been going on. So it's Richard Lenski's lab. It's at Michigan State University. And Lenski has really had some really dramatic contributions and input into the field. So let's see if we can find that. Lenski experiment, Michigan State. He has his own website. So here we go. And him and all of his grad students and postdocs have done thousands and thousands, right now more than 60,000 generations of this long experiment. Okay, so let's just highlight just a couple things because there have been dozens and dozens of publications and lots of information that's come out of this. All right, so the question is, what happens when we take a bacteria that is adapted to a certain environment and put it in a new environment? So you could do this with bacteria with temperature, for instance. If the optimal temperature for E. coli bacteria is right around human body temperature, what happens if we put them into a cooler environment where they can still survive but don't do as well? Or we could do it with a food source. What happens if you give them a suboptimal food source that they can still survive on, but they just don't thrive on, they don't do as well? And so they've done this. They've done this in the lab where they put them into a suboptimal environment. And what they see by measuring fitness, so this red line is a measurement of fitness, and you can measure it via cell volume and then kind of make an adjustment for how fit they are. And what they saw is over time that fitness increased but reached kind of a plateau level, right? So this makes sense, right? You put an individual into an environment that they're not perfectly adapted to, and then over time they will adapt to that environment. Now remember, this starts with a single bacterium. So in essence, there's no diversity when you begin. And so if there was no possibility for change, that bacteria would stay at a constant level. And so obviously there's change here going on. And again, remember, where does that diversity come from? Well, there have to be new mutations. And so because bacteria have a fairly high mutation rate, after maybe just a couple dozen generations with thousands and thousands of bacteria, we have one that has a new mutation that allows it to take advantage of its new environment a little bit better. And then we have a second mutation, a third, and a fourth, and a fifth, and many, many mutations together allow them to adapt to their environment. Now the question is, if we run this experiment multiple times, which we can do with these bacteria, do we have the exact same end result? So I want you to think about that. Think about what you've learned about the nature of mutation, natural selection a little bit too. But natural selection is if it's good, it's going to be kept. It's going to be now common in the environment. With bacteria, that can happen in just a few generations. If we have one bacteria that is substantially better than all its others, it will outcompete them, and soon every bacteria will be descended from that newly changed and better adapted bacterium. Okay, so would you expect the exact same results? Well, here are the results of that experiment. So here we have 10 or 12 different experiments measuring fitness over time. And notice that although in general we get the same result once we adjust, this is just cell volume, and then we make some adjustments for overall fitness that's based on generation time and a couple other things. here is the result of the experiment. And although the general trend is the same, the end result is slightly different. And this is because mutations are random. And we might even have different mutations. There might be some that are the same or at least give the same end result. But different mutations occur in different populations of bacteria. And so we end up with the same general result, that they adapt to their environment. But some of them have done it a little bit more quickly or a little bit better than others. And this is because of this random nature of mutation. So this is just bacteria, but we could start thinking about this in longer terms. What would happen if we were able to hit the rewind button in, say, the history of life on Earth? Let's go back 65 million years, exact same environment, and let's have that mass extinction event that ended the Mesozoic era, began the Cenozoic era, which is the current era that we live in. Would we have the exact same result? Now, the results of that extinction were that reptiles, diapsids, right, still were around, but were not as dominant as they were afterwards. And replacing them were mammals in many niches. Some diapsids still did well, like birds, crocodiles, lizards, and snakes, do all right, and they thrive afterwards. But many of the diapsids, the pterosaurs, the mosasaurs, most of the dinosaurs, plesiosaurs, all of those organisms go extinct. So if we're able to replay that experiment, quote unquote, right? It wasn't really an experiment, it was a massive cataclysm. Would we end up with the exact same result? Would mammals still dominate? And the short answer is we don't know for sure, but almost certainly we would see some differences. So are there certain things in evolution that are inevitable? And the answer is probably there are general trends that we would see, but we might be able to get there via different mutations. And so we really don't know the answer in detail to this, but in general we could say if we were able to hit the rewind button, we would probably see some differences, maybe even some substantial differences. And really we don't have an experiment we can perform. The closest we are going to be able to do this is to discover life on other planets, which in a way is kind of like doing a separate independent experiment. Right? If we look at life on Earth, we really only have one big massive experiment in the evolution of life, and that's our Earth. And it's incredible and diverse and amazing, and we can study a lot about it, but it really in the long run is just one big experiment with many, many sub-experiments, right? So we can look at, what happens when we get a new island? Do we get the same thing every time? And we have different characteristics of each island. We have different species founding it, and so it's really not a very similar, well, in some ways it's similar, but it's not an exactly similar experiment. But we could look at it at different planets if we are ever able to assess life on other planets. So questions like, are vertebrates inevitable? If we go to another planet, do we find vertebrates? What about DNA? Is all life on Earth DNA based? And if so, are they going to use the same genetic code or a very different genetic code? And the short answer is we don't know for sure. We're probably going to see some things that are similar because there's a limited number of ways that you can solve a problem. And so we might see vertebrates on another planet. It's very unlikely that it will be, I always laugh when you look at Star Trek, right? So what do all of the aliens, especially in the early days, what do the aliens in Star Trek look like? Right? Vulcans look like humans with pointy ears. Same thing in Star Wars, right? These alien races just look like weird vertebrate humans, maybe not even that weird, right? And so that's assuming a remarkable amount of convergent evolution, that if we reran the experiment, we would get a very, very similar result. And we don't know. Maybe that's the case, but we won't know until, maybe this will not happen, maybe even not in our lifespan, until we can find life on other planets. It's likely it's out there just based on statistics and the number of planets that we now know are out there in the universe. But we don't know what life will look like, if it will be similar or very, very different. Now, one of the other outcomes of this long-running bacteria experiment is the importance of antagonistic pleiotropy. Now, that's a long word, so let's break it down. Pleiotropy is where we have one gene with more than one phenotypic impact. So one gene, 2, 3, or 4 jobs, right? That gene does multiple things in that organism. And antagonistic simply means that there's some sort of a conflict there. So if I have a gene that allows me to reproduce more quickly, but it maybe makes me less able to digest food, or vice versa. Maybe I can digest food better, but I can't divide my cells as quickly. Then whether or not that will be an adaptation will depend on the force on both areas. So it's a trade-off, right? So let's say I can reproduce more quickly, but I can't digest my food as well. Is that an advantage? Well, it's going to depend on the environment, and it's going to depend on how much of an advantage-disadvantage trade-off we have going on. And we see antagonistic pleiotropy all the time. So we'll come back to that. It's not just present in bacteria, it happens in all organisms. Often there is a genetic trade-off. Something that is an advantage might also provide a downside. But if the advantage outweighs the downside, it will still be selected for overall. So let me give you an example of some other antagonistic effects that put constraints on evolution. And there are genetic levels here, but this is really complex morphological traits. So this is an example of a physical constraint that's similar to that antagonistic pleiotropy. Organisms that have two eyes have very, very good potential for binocular vision, but only where those two eyes overlap. So that's basically how we judge depth, how we judge distances, is we get an image from one eye, we get an image from our other eye, and then our brain interprets that image to say how far away things are. And particularly with movement, it's very, very effective. But if you only have two eyes, you have a limited range of vision. If you want good binocular vision, your eyes are going to be right in the front of your head, but you have a large blind spot. Ostriches, so owls, have very good, and they've adapted to an environment where they need good binocular vision. Ostriches have the eyes on the side of their head, which means they only have a very narrow range of binocular 3D vision, but they have a very small blind spot. They have a very wide field of view. So, which is better, to have eyes on the front of your head or to have eyes on the side of your head? Now, hopefully, again, I didn't trick you, right? The answer is, well, it depends. It depends on the environment. So ostriches are adapted to an environment where they need to be constantly aware of what's around them and be on the lookout for predators. And so they have adapted to this very wide field of view, but the trade-off is bad binocular vision. Owls, on the other hand, because their predators need very good binocular vision, they hunt at night, they have very good night vision, they use both hearing and vision to grab their prey. And so they need really, really good binocular vision. And so the trade-off is they have a very big blind spot. Now, what we see is when there is antagonistic pleiotropy and there's a trade-off, often the downside of that trade-off will be alleviated when a new mutation or a new variant that comes into the population allows for some alleviation of the downside. So let's look at how that happened in owls. Owls have incredibly flexible necks. So by sitting in one place, they can turn their neck more than 180 degrees, which is basically all the way around, right? Kind of like, what was The Exorcist, right? That movie about a demonic possession where the main character turned her head all the way around. They can't quite go all the way around. And so that is an adaptation that relieves some of the constraint that was placed by this initial trade-off. Now, jumping spiders are the same way, that they need really, really good binocular vision. They're also predators. They can't move their heads, however. And so we have a different solution to the same problem. They have evolved eyes essentially on the sides, but almost to the back of their heads. So they have really, really great 3D vision by having multiple eyes. So different ways to alleviate this. Now, there are always going to be limits on how much organisms physically can do. So, for example, if it's really, really good to be large, there's going to be selection for larger size. But there's some constraints on how big you can be, because that puts an incredible amount of strain on the joint. So this is a sculpture on the left here by Salvador Dali. If you know much about Salvador Dali, he liked to do kind of extreme, surreal things. And so if we really had an elephant like this, it would crush its legs. There's no way that the weight with modern bone strength and even with the strongest possible biological legs, you could not support that amount of weight on those skinny legs. And so if you're very large, there are going to be constraints on you that are not imposed on very small organisms. But at a small size, those constraints are different. So daddy long legs, if we blew a daddy long legs up to the size of a human or an elephant, thank goodness they don't get that big, right? But if we did, it wouldn't be a problem, because at that size scale, their body would collapse under their own weight. They could not support their weight on these legs. And organisms can adapt, and secondary changes can make up for the original physical constraint. So horses have this one single digit. Remember when we talked about the evolution of horses? This is their femur, their tibia and fibula, and so that right there is the equivalent of their ankle. And then we've got their wrist bones and their phalange, and they're running on a single phalange. And so one of the reasons why horses have so many injuries is they've kind of been pushed to the limits of their physical constraints for their size. And so evolution will work to make things better, but sometimes that comes with trade-offs. Those trade-offs can sometimes be partially solved and alleviated by secondary mutations, but still there's not going to be a perfect solution in many cases. And so that is often a misconception about evolution. Evolution doesn't find the perfect solution. It doesn't solve all problems. All it does is take the diversity that's present in the environment and pick what's going to be best. And if it's better than it was before, great, then organisms adapt and they evolve into their new environment. Sometimes a new mutation occurs and it's better than what was there before, and so then that would be selected for. But not always, just by random chance, do we find the perfect selection. So we'll look at examples where we have adaptations that are good, but maybe not the best that we can in a later video. So to end this video, this discussion, there is limited variability. All of that variability is brought in by natural or by mutation, and the natural selection works on it to decide which is going to be stay and be a beneficial trait, and which is going to be removed because it's negative. Now, sometimes there is diversity that is really not good or bad, and so it's also a misconception to look at any trait and say, oh, it's got to be either an adaptation or detrimental. Some of it is just diversity. So, for example, in humans, is it an adaptation to have brown eyes? Is it better to have brown eyes, green eyes, hazel eyes, blue eyes? What's the best trait overall? And probably the answer is, maybe we could experimentally test this, but probably the answer is, it doesn't matter, right? Is it better to be A blood type, B blood type, or O blood type? Does it provide you an advantage if you're one or the other? And the answer is probably not one or the other. So there's lots and lots of diversity that's just popped up. Natural selection doesn't work on it to either make it more common or to remove it. And so we end up with lots and lots of variability. And sometimes then an environmental change will come along and make a trait that was just variability, turn it into an adaptation. And we have a word for that. So an exaptation is when an environmental change makes something that was there before and wasn't good or bad, it was just neutral, but suddenly it makes it an advantage. So if suddenly there was a huge advantage to having brown eyes, then we'd see a shift towards more and more brown eyes in the human population. Now that's not, I can't think of any advantage, maybe there will be something in the future, but I can't think of any advantage that might occur. So let's use a real example of an exaptation. The vertebrate skull, because of the way it evolved, is put together by many, many bones developmentally. And these bones grow and then fuse together. Now, in most animals, that doesn't make a difference. If the skull all grew from one piece, or if it grew from many pieces, it really wouldn't make a difference in their survivability. But in humans, that has provided an advantage to us and alleviated some of the constraint on brain size. So there's been very strong selection in the human lineage to have larger and larger brains. But there would have been a much lower constraint on the size of our heads if our skulls were built from one piece. What actually happens during development is the infant skulls grow from different bones, and those bones do not fuse together until a year and a half or so after they're born. And so what this means is head size can get very, very large, and we've kind of been pushed to the extreme of this because of the size of the birth canal, right, where basically natural selection has pushed human head size to as big as it can get and still get through the birth canal. And that's been a little bit larger because the skull is flexible during birth. And in fact, you'll see this, particularly after very strenuous births, that the head of the baby is somewhat deformed. Sometimes you get a little bit of a cone head. And usually that will resolve on its own self and kind of go back to normal. And then when the baby's about 1.5 years old, the soft spot and the fissures between the skull plates will fuse together. So that's an example of an exaptation. There are many, many others. If you want to look at examples, that's fine, but just know that one for this class. So an exaptation is diversity that was already present in the environment, and then an environmental change means that suddenly one of those traits is going to be a big advantage and then therefore be selected for.